Hello, my name is Katie Christensen. I'm a junior studying agricultural and medical biotechnology as well as violin performance. And I'm conducting research with Dr. Kendall Corbin in the Department of Horticulture. I'm willing to bet that at some point in your life, you've taken antibiotics. But did you know that by 2050, death by antibiotic-resistant bacterial infection is expected to increase from 1 million deaths per year to 2 million deaths per year, meaning the antibiotics that you and I take are becoming less effective. For the past 75 years, we have used antibiotics to treat bacterial infection, but have stopped giving time and money to create new antibiotics and continually overprescribe existing ones. This has led to a point of crisis with antibiotic resistance. My research focuses on the first of these issues, creating new antibiotics, specifically from an underused source. You see, most antibiotics come from soil samples. However, my project involves looking at maize leaf surfaces to identify the bacteria and test them for antibiotic properties. You can see at the top of my methods section that sterile swabs were used to streak maize leaves and then were transferred to 4 general types of agar media, which I'll refer to as media types 1 through 4. Now, there are many different types of bacteria growing on these plates, so they had to be isolated from each other to result in 240 isolates. The last 2 steps in this first set of pictures shows that I used a lab technique known as polymerase chain reaction, or PCR, to make billions of copies of the 16S rRNA gene from each of these isolates. This gene is a unique gene in bacteria that is used as a bacterial barcode to generally identify the species. So after creating my billions of copies, I was able to sequence the DNA of these genes and then identify my species. I found many different types of species of bacteria, but one group that stood out to me in particular was Pseudomonas. Pseudomonas is a genus of bacteria that has been known in the past to create clinical antibiotics. Thus, I decided to test my Pseudomonas isolates for antibiotic potential. To do this, I grew my Pseudomonas isolates alongside 5 relatives of known antibiotic-resistant bacteria on separate plates. If my Pseudomonas isolate produced what is called a ring of inhibition, where it outcompeted the harmful relative bacteria immediately surrounding it, it has antibiotic activity. You can see a clear example of a positive ring of inhibition at that top plate right there. That bacteria to the right is my Pseudomonas isolate, and there's a distinct ring around it where it has killed off and outcompeted that harmful relative bacteria. In contrast, that same Pseudomonas isolate, when put against a different relative bacteria, created no ring on this plate and thus had no antibiotic activity. 19 out of my 27 Pseudomonas isolates produced antibiotic activity, with those from the 3rd type of media having the lowest percentage of antibiotic activity, and those coming from media types 2 and 4 having the highest rates of antibiotic activity, with all 100% of their isolates having antibiotic activity. You can see this in the second graph of my results section. Additionally, that 16S rRNA gene we talked about earlier, well, when I compared that gene for my different Pseudomonas isolates, I found that the Pseudomonas isolates that came from the same type of media were more closely related to each other. Now, this data is incomplete and is still in progress. Based off of these preliminary results, I have identified 3 isolates, and from each of the 3 types of media with higher rates of antibiotic activity, so 1, 2, and 4. If you look in my future directions section, that graph will show you that these 3 isolates I have selected have also outcompeted all or almost all of the 5 relatives of antibiotic-resistant bacteria. My next steps will be to sequence the full genomes of these 3 isolates so that I definitively know exactly what species and strain they are. Additionally, I will chemically extract the antibiotic compounds they produce so that I'm one step closer to clinical antibiotics. Antibiotic production is necessary to counter antibiotic resistance. My research shows that sources outside of soil can and should be used for sources of antibiotics in this fight. If we take this problem seriously, we can slow down and possibly counter the effects of antibiotic resistance. Thank you so much, and I'm ready for questions.
Why do you care about the media types?
So the media types are very general, but they're just a good way, they're slightly selective, so they're a good way to show that while many types of Pseudomonas grow on this type of media, they didn't necessarily grow on this type of media. So it is just an easier way to compare all the Pseudomonas isolates.
So you're just trying to get differentiation between the species that you're getting?
Yeah, exactly. It's a good way to sort them out and be able to see, okay, why do these behave the way that they do? Well, they come from the same media type and they're more genetically similar.
If you're taking your samples off the surface of the maize leaf, is there any significance to the fact that it's maize leaves, or is it just the environment that's, resting on the surface of the plant?
That's a great question. If I'm understanding it correctly, you're asking why maize leaves, why not just anywhere other than soil?
Yes.
Okay, that's a great question. It can be anywhere other than soil. It's just not very common to do it apart from soil samples. So this was bacterial species that I had access to, and I thought it would be a good place to start. But I'd love for this project to encourage more antibiotic testing from other sources, other plant sources, other general surfaces, airborne like bacteria.